Okay. Good afternoon. Thank you for joining us for Astardi Medical's webinar hosted by Dr. Nicholas Embleton, Nutritional Status and Gut Health in Preterm Infants. What is it and how do we measure it? I'm Tammy Jansen, co-founder and CFO of Astardi Medical. For those of you who don't know us, Astardi Medical is a precision nutrition company providing digital solutions to support nutrition and feeding practices for preterm infants in the NICU. We're hosting a series of educational webinars this year, all focused on the growth and development of preterm infants and the importance of feeding, nutrition, and gut health. I'm thrilled to announce Dr. Catherine Gregory will host our next webinar on September 17th. Dr. Gregory has dedicated her research and clinical career to improving health outcomes for preterm infants and their families, focusing primarily on GI diseases, nutrition, the microbiome, and its role in gut health for these fragile patients. Please mark your calendars and join us for a presentation by Dr. Gregory on building a better baby biome, caring for our babies and their bugs. So let's get started. We'll have plenty of time for questions at the end. Please feel free to post your questions throughout the presentation in the question box. Just scroll down and you'll find it at the bottom of your screen. We'll compile the questions and get to as many as we can. So I'm pleased to introduce our esteemed presenter and expert in the field of neonatal nutrition and gut health, Dr. Nicholas Embleton. Dr. Embleton is consultant neonatal pediatrician and professor of neonatal medicine, Newcastle upon Tyne, UK. He is an elected member of the ESPGHAN Committee of Nutrition, coordinates the UK-based Neonatal Nutrition Network, and has over 175 peer-reviewed publications, in addition to numerous educational articles and book chapters. I'll now turn it over to you, Dr. Embleton.
Great. Thanks very much, Tammy. I'm just going to upload my slides and make sure I can get those going.
Yep.
Okay. You can see the slides okay?
Yep, we can see them.
Okay, I'm off. So thank you very much to Astardi for inviting me. I'm delighted to spend the next 40 minutes or so speaking about nutritional status and gut health, provide an overview to the area as I see it. I'm aware that we're quite a broad audience today, so there's a mixture of medics and nurses and nutritionists. So I hope I've pitched it at the right level to everybody. I hope I can keep you entertained. Somebody's chipping in there saying they can't hear. I'm assuming, Tammy, you can hear me okay, so maybe... Okay, so maybe Rich hasn't got his headphones. I'll keep going, and people I guess, will catch up.
Well, yep, okay.
Okay, so just a few conflicts and disclosures, just so you guys know where I'm coming from. I've been a neonatal clinician for about 20 years, attending in the UK. I've received research funding from National Institutes for Health Research, which is the sort of main body for funding medical research in the UK, Medical Research Council, but also various charities who've financed some of our important studies. Important to note that I have conducted a study with Prolactic Biosciences, who make human milk fortifier, and a known Early Life Nutrition, who produce formula. And I've received lecture honoraria from Nestlé Nutrition and from Baxter, but I don't hold any shares or stock options and don't have any other relevant conflicts to disclose. And I wanted just to point out a few of the key biases that I have. The first is that good nutrition in the first 1,000 days, that's the 9 months of pregnancy and the first 2 years of life, is critical both for short and long-term outcomes. Most of the preterm infants that we look after, I think, do not receive the nutrition they deserve and that they need, and that all of us could do better to improve the nutritional status of the babies that we're looking after on our neonatal units. A little bit about the background of where I come from and what we do. So I'm based in Newcastle, which is in the northeast of England. We run a series of collaborative trials, some of which you can see on the screen here. So we're involved in running the big lactoferrin ELFIN trial, but also the SIF trial. We're doing really a lot of work around the microbiome and metabolome in the gut. We've got specific projects looking at the minute at secretory IgA in breast milk, looking at bacteriophage, i.e. viruses in breast milk and how these may be transmitted. We've done some work looking at human milk oligosaccharides. We're very interested, of course, in NEC and the immunological processes that might be underlying it. And we work with Chris Stewart, that you can see in the bottom left of the picture here, who has developed gut enteroid models. And we've developed these using stem cells that we've retrieved if babies have had operations for NEC. So we get parents' permission to retrieve a small bit of tissue, and then we can grow these mini gut organoids in the lab. We've got projects looking at long-term metabolic outcome, and Janet Berrington, who's in the bottom left there, coordinates our large neonatal biobank. And within that biobank, we hold stool, urine, salvaged blood, breast milk, and some tissue specimens from over 1,000 infants. So we've got a huge repository here for doing our work. And just finally, Newcastle, beautiful place in the northeast of England with some lovely castles and beaches and nightlife and beer, and a football team who never win anything, but they're good fun to watch. Okay, so what am I going to talk about in the next 30-40 minutes? I want just to give you my spin on what I think nutrition is. I want to highlight the importance of brain growth and nutrition in preterm infants. I then want to give you my take on what nutritional status means when we talk about babies or even adults being in good nutritional status. What is it that we really mean, and actually what are the measures we can use to assess it in clinical practice? And then talk a bit about gut health and how that might fit in. And just to link there to a couple of websites, we have our own research group website, neonatalresearch.net, which describes some of our research activities. So you can go there. I put all the slides from our various lectures on that. So if you want to look at any of these slides, they're free to view there. As Tammy said, I also help coordinate the Neonatal Nutrition Network, which is a group of clinicians across the UK. We've been meeting for over 15 years now. We run a series of educational meetings, and we act as a sort of hub to discuss research studies and plan them. Okay, so achieving optimal nutrition in preterm babies, this is what sometimes people think it is, that the way to do it is you just stick the baby on the scales, go to the shelf, get the big diet book out and work out how many nutrients the babies need, multiply the nutrients by the weight, put those nutrients into a solution, ideally milk, but if not, something parenteral, give them to baby, and then watch them grow and turn into somebody like Einstein. And if that was all we needed to do, this could be a really short talk. But all of you will appreciate that actually knowing the precise quantity of nutrients that a baby might need to grow doesn't actually achieve good growth and good nutritional status in the babies we're looking after on the NICU. So what is nutrition? When I came to this area many years ago, I got very focused on the nutrients, and I dare say many of you do that as well. But for me, nutrients are just one element of overall package of nutrition. So within nutrients, we of course have all your building blocks, your macronutrients, your micronutrients. But in addition to the nutrient aspects, of course, we're now increasingly aware of the functional components. So these are particularly human milk oligosaccharides, but also growth factors, cytokines, enzymes that might be there present in, particularly in breast milk, obviously. We're increasingly aware of the importance of microbes, both those acquired and promoted by breastfeeding and breast milk, but also the wider environment. And of course, there's a lot of work going on trying to define the role of probiotics in preterm infants. And then the last element of nutrition, I think, are the technical or the socio-behavioral aspects. Again, I think these can be overlooked. Most neonatal practitioners are very aware of the kind of debates around bolus versus continuous feeding, but also the importance of skin to skin in promoting breastfeeding, but also now sensory aspects. You know, how does this food taste? How does the milk taste in the mouth? And what are our own sets of beliefs and behaviors that may promote breastfeeding? So if we want to really approach nutrition, we need to take a holistic approach and consider all of these four elements when we're planning our care. Now, brain growth in humans is really very different to most other primates. When I started looking at this area, I assumed that human brains would grow just the same as other primates. But humans really are very different. In the chimpanzee on the left, the rapid period of brain growth happens in the second and then starts slowing in the third trimester, so that monkeys are born with a relatively large brain. Whereas in humans, we have this difficult compromise between having to grow a huge brain, but also then having to be delivered by mothers who walk around on two legs. And so a huge amount of brain growth happens in the period both before and after term. So that between 24 weeks gestation and 2 years of age, humans acquire about 90% of their final adult volume. So massive amount of brain growth happening in the period in which we as neonatologists look after these babies. And in humans, really, it is all about the brain. Human physiology, placental physiology, transport of nutrients, everything that's in breast milk is all there to promote and support a very big brain. So nothing about how nutrition and nutrients evolved to stop us getting diabetes or to stop us getting fat or having blood pressure. That's really not that relevant. Everything is about promoting growth of the brain. And what I show on the left here is a picture of a rhino and a rhino skull. And you can see here that I picked out in blue the size of the rhino's brain. So what you see is this great big head, but actually a relatively small brain. Whereas in humans, the brain occupies the entire cranial cavity. So we have huge brains compared to most other animals. Here's another way of looking at it. If you plot out body weight on a logarithmic scale on the x-axis at the bottom there, and brain weight up the y-axis, what you see is that most mammals plot out somewhere on that line. So a rhino and a shrew in the bottom left corner have brains that are slightly smaller on average compared to other mammals. My own pet dog, they're pretty average intelligent, despite what my children might say. But humans are off the scale. Humans have a brain that's about 10 or 20 times as big as any other mammal. And we have to appreciate the importance of adequate nutrition in growing that brain. Now, when I talked about brain volume being acquired between 24 weeks and 2 years of age, of course, whilst that parenchymal mass is being acquired, there are lots of different processes going on. So throughout pregnancy, neuronal migration is very active, but then is largely absent after term. In the second and third trimesters and in the immediate few months after term, apoptosis, programmed cell death, is very active. Synaptogenesis kicks in the third trimester, but then carries on for the first 1 or 2 years of life. And myelination, although this starts at third trimester, may not be complete until about 20 years of age. So lots of different processes going on. And there's now increasing evidence that all of these processes are under the control of growth factors, particularly IGF-1. And again, lots of recent work in the last 5 or 10 years showing that nutrient intake, dietary intake, modulates levels of IGF-1 in babies. It's not the only thing that modulates IGF-1, but it's one of the key modulators that we can change. So we're now starting to understand that there is a direct link between the nutrients that we might feed babies and how you may be promoting or inhibiting brain growth in preterm infants. So how might nutrient deficiency damage the brain? So let's superimpose 3 hypothetical nutrients on top of that schema of the different neuronal processes. And the impact of a nutrient deficiency on the outcome will depend on the amount of deficit for that nutrient, the type of nutrient, when the deficit occurs, and for how long it lasts for. So in A, you can see a nutrient where there's an important deficit, but only for a relatively short period of time immediately after birth. B is a different sort of hypothetical nutrient, which may not be critical in the first few days, but becomes more important when the infant grows. And there may be another nutrient like C, where the lack of it is important even in the post-discharge period. So A might be something giving you a different profile in terms of the processes it's affecting to nutrient C. So example of A would be macronutrients. We actually know, and I'm going to show you a little bit of data, that it takes time to establish macronutrient intakes. Once we've got them established, as long as the baby keeps tolerating the feeds, it's relatively easy to meet macronutrient demands. But A is an example of an important deficiency that is often overlooked. B might be something like iron. You actually don't need very much iron the first couple of weeks as a preterm baby because you're born with plenty of stores and often you're getting transfusions. But if people forget to start iron, this could harm brain development after the first 2 or 3 weeks. And nutrient C might be something like DHA or choline. Again, emphasizing the importance of nutrition in the post-discharge period. Lipids, clearly a key component of all your neuronal tissue. We know that intravenous lipid solutions are probably suboptimal, although some of us are using fish oil and products like SMOF. These may not be the appropriate balance and quality. We also know that maternal diet may be lacking in DHA, and that breast milk levels of those mothers may also be inadequate. A recent study came out just in the last couple of weeks, though interestingly showed that giving mothers DHA supplements doesn't improve outcomes and may actually increase the risk of lung disease in preterm babies. So this is a very complex area where we need to appreciate the relationship between nutrient deficiency and phenotype is going to differ. So lots of different nutrients, changing diet from parenteral to enteral nutrition, lots of developmental stages, and multiple mechanisms. I can't ask you if you know who this guy on the left is here, but this is Egan Bernal, who's a Colombian cyclist who won the Tour de France last year for the first time. So an amazing young cyclist, and this guy cycles up and down mountains for 3 weeks non-stop. It gets up in the morning, they cycle up and down for 6-7 hours, and then they get up the next day and do it all over again. They weigh typically just under about 70 kilos, and they're consuming 7,000 kilocalories a day. So this guy is burning 100 kilocalories a kilo a day. Just imagine how amazing it must be to eat 7,000 calories a day without getting fat. So I think we're all a little bit jealous. But that's the energy required to cycle up and down mountains for 6 hours a day. Compare that to what we know preterm infants need. We know that most of our small preterm babies are going to need a caloric intake of somewhere between about 120 and 130 kilocalories a kilo a day. So our babies have an energy expenditure that is about 20 or 30% greater than the most energy consuming exercise known to man. I think that's really quite incredible. And when you think about where that energy expenditure is partitioned, you realize that over half of that, half of those calories are consumed by the brain. So very easy to start to understand why failing to provide adequate macronutrients to these babies may result in worse brain outcomes. So stores are limited and demand is incredibly high in these preterm babies. So how much do babies need, and what are they actually composed of? So people say, you know, how do you make a baby, or what's a baby made of? So some seminal work conducted by Eckhart Ziegler and others back in the 1970s looked at the different body proportions in fetuses. And what you see here up the y-axis is the percentage components of the body, and then gestation along the x-axis. What you see is that minerals only account for 1 or 2% of weight. Lipid counts for virtually nothing at 24 weeks, but around about 15% at term. And protein is fairly static throughout the third trimester at about 10 to 12%. So 90% of a baby at 24 weeks, 90% of the weight is actually made up of water. So when you're looking after these babies weighing only 500 grams, you're looking after an organism that only consists of 50 grams of solid matter. I think that's really quite incredible. So what does that 50 grams look like in terms of energy? So in the UK, we have a chocolate bar, Mars bar, which interestingly weighs exactly the same as a 500-gram baby. So they both got 50 grams of tissue. If you look at the total available energy in that baby, that's 250 calories. That's everything that baby has together. Clearly, the baby couldn't use all that energy, otherwise there'd be nothing left. So the energy stores, although they aren't strictly stores, but the the available energy for that baby at the point of birth is probably less than 50 calories. That's all that baby has between it and death. So how might nutrition impact on the brain? I think there are lots of different mechanisms. I think the first two would be making sure that you have enough macro and micronutrients to actually accrete your tissue. Next, you have to have sufficient energy to drive the system, so particularly your carbohydrates, your lipids, but if you're not providing enough of that, then you need protein, or you burn your protein. But you may also impact on brain development by not providing the right types of signaling and growth factors. So this is particularly the proteins and phospholipids associated with milk fat globule membrane, but also IGF-1 that appears in breast milk, epidermal growth factor. A whole range of nutrients impact on gene expression. Folate, B12, iron, DHA, and choline are all known to exert effects through epigenetic mechanisms. And then the final mechanisms might be microbial mechanisms, prebiotics, HMOs, lactoferrin probiotics that may improve gut health and stop you becoming unwell, particularly also then finally breast milk. Because breast milk, we know, reduces the risk of NEC and sepsis, and anything that reduces NEC and sepsis is going to improve brain outcome. So this is a photo from the stabilization area of our NICU that I took a year or two back, and you can just see the complexity of equipment and monitoring required to look after preterm babies. And every day we do rounds and we think about all these different factors. You know, what's the level of bilirubin? Should you put a central line in? How much fluid? Which inotropes? What saturation level should the baby be at? How long do you continue the antibiotics for? What temperature to keep the baby at, urine output. I mean, these are the things that are discussed on rounds every day. And when you stand and listen to people's rounds, you'll see that not only is NICU care very complex, but it tends to be focused on cardiorespiratory monitoring. And when you look at all that equipment there, really none of that is there to monitor nutritional status or nutritional inputs. So there's a real lack of focus generally on nutrition on the neonatal unit. So what's the consequence of that? Now, this is work that I did actually now more than 20 years ago, where I looked at macronutrient intakes over the first 2 weeks of life. Of course, we're now recommending protein intakes close to 4 grams a kilo a day, but back then we thought that if we got to 3, we would be doing pretty good. So those yellow bars represent the 3 grams a kilo that babies should get on each of the first 14 days of life. We were pretty good at starting them on feeds. We, even 20 years ago, got the first feed in with the first 1 or 2 days, and we reached full feeds by maybe day 10 in most babies. So we were feeling pretty confident that we were doing a good job in terms of starting early feeds. But then we actually looked at what the babies were actually getting, and in green, the green bar depicts the actual protein intake. On the first day, we didn't give these babies back then any PN, and when we started PN, we used to build it up really slowly. So the green bar represents what they're actually getting compared to the yellow, which is what's recommended. And then the red bar represents the difference between the two. And what you can then depict is this accumulation of a nutrient deficit that just keeps on growing and growing. And so although by 2 weeks of age, it's slowed off, you can see that by then babies have accrued a cumulative deficit of protein around about 18 grams a kilo. That's roughly equivalent to not giving the baby anything for 5 days. And if you repeated that using 4 grams as a recommended intake compared to 3, it would look even worse. Now, I know we're a lot better on that, but this happens without people seeing it happening. It just goes on without you recognizing it. And so whilst we think we're doing a great job in starting these babies, we're actually most of the time severely malnourishing many of them. And as I say, part of the reason is that our monitoring is all focused on cardiorespiratory care. And I often wonder how good it would be if we could design a monitor that somehow provided us with the same feedback you get for the cardiorespiratory system. If we could somehow integrate the little barcodes you get on all your PN supplies and formula milk, you could then just multiply that by the amount in the syringe pump and get your answer. So this is what I did. I made a little QR code to go on the different solutions that we give the baby. I'm going to build a little infrared scanner that will just scan the barcode, multiply that by the infusion rate in the pump, and then build my little monitor here. So here I've built a little monitor that will give you a printout of energy and protein. And if you want to go to Amazon, you could buy that for me now. But maybe somebody else will invent a monitor like that that will give us this feedback that at present we don't have. So I've started to tell you that nutrition is much more than simply nutrients. We need to consider microbial, functional components, and all the kind of techno behavioral aspects as well. We need to really focus on this one brain for life and the multiple overlapping developmental stages, and that the impacts of nutrient deficiency depend on the amount, type, timing, and duration. Furthermore, we all recognize, I think, how complex neonatal care is and how common poor nutritional status can be. So how do we assess nutrition? How do we work out whether these 4 bits on the left, the nutrients, functional components, microbes, and social elements, are putting this baby into a good nutritional status or poor? And what is nutritional status? When we talk about it, really, what do we mean? And how do we actually assess this in clinical practice? So if I asked any of my fellows, I don't know if any of them are listening, they're probably down the pub drinking beer whilst I yet again stay at work, but they look at me like this, kind of, what are you asking about? What is it that you want me to do? If I said to them, go off and assess respiratory status, they would go and they would look at respiratory rate, cyanosis, recessions, they would probably listen to the chest, look at the oxygen saturations, they probably would measure blood gas. And similarly, if I said, assess cardiovascular status, they would do that and they would look for cyanosis and heart sounds and blood gases, and they probably would pull out that very clever echo machine that gives you lots of pretty patterns, but nobody really knows whether it helps the babies or not. But when it comes to nutritional status, everybody looks a bit blank. If you stand there long enough, then people will weigh the baby and measure them, and if you're really lucky, they will plot them on a growth chart. And if you're really, really lucky, they'll make the plot in the right place. And then somebody might suggest some blood tests, but they're not really quite sure what blood tests they should be doing. So what do we mean by nutritional status? Well, the starting point is to appreciate that nutritional status is dynamic and multidimensional. It's perhaps easier to start from adults and understand how we might do this in adults, or how we might define it in adults. And this came from a course I did on human nutrition many years ago now. So the credit's to them, not me, but they say you should think of nutritional status as what you eat, i.e. what is your typical diet and exposures to different types of foods, what you are, i.e. what is your body composition, how much lean mass, how much fat mass, what is there, and what is it you do, what is your typical requirement for physical activity, but also functional activity of all the cells, tissues, and organs in your body. So there's no single measure of nutritional status. There's not a single test or thing that you can send to the lab or measure. And in preterm infants, expanding this concept can be quite difficult. We know that nutritional requirements vary depending on maternal health, growth restriction. It's going to be different in a baby recovering from NEC compared to a baby who didn't have NEC. It's going to be different in a baby with lung disease. How do we put it all together? So it's important, I think, to appreciate the life course nature, that what happened in the past, so the nutritional status in the past, determines your metabolic capacity in the present, and that will determine your nutritional status in the future. And lots of you would easily be able to think of all the factors that might fit into that model. So of course, there are going to be genetic and epigenetic factors in the mother, aspects of maternal diet, drug exposures, differences in placental growth and IUGR, but also the past neonatal history. The present aspects consist of your metabolic capacity, your ability to handle glucose, lipid, or amino acids, your current body composition, what diseases are going on, and what your typical diet looks like. And then the future, the things that we've touched on before, so particularly the brain in terms of behavior and cognition, but also metabolic outcomes and size and growth in later life. So this is the little schema that I've tried to promote for people, saying nutrition assessment as easy as ABCDE. And I'm going to go through these elements one by one and just show you how I would approach it. So anthropometry, obviously weight, length, head, plot it on a chart, bloods, body composition, clinical aspects, dietary aspects, and then the evaluation. So I'm just going to deal with these quickly one by one. Anthropometry, as we heard from Bill and Tanis last week, very important. Babies need to be weighed probably at least 3 times a week, maybe more often. Head and length, ideally weekly. plot them correctly on a growth chart. We could spend a lot of time arguing over which growth chart we should use, and I'm not going to get into that debate today, but we all need to be aware of the limitations, the reference data of the growth charts that we're using. And of course, we're talking about growth here and not size. So growth is really important, but it's really important because it's a measure or a marker of nutritional status. It's not actually an outcome in itself. It's not a functional outcome, but clearly it's a very good indicator of whether your nutritional status is good or poor. And really what we're trying to do is achieve good nutritional status rather than achieve a specific rate of growth. So we have to assess that over a period of several days and recognize that all babies are different and there's going to be no one size fits all. I'm not going to dwell on what I think is normal or abnormal growth. I've picked out a few rules of thumb that I tend to sort of remember and use on rounds. Weight gain under 30 weeks is somewhere between about 18 and 21 grams a kilo a day, but it's a bit slower after 32 weeks. Weight gain in grams per day varies a lot, huge variability. And then looking at head circumference gain just under a centimeter a week and length gain around about a centimeter a week. But measurement error, particularly in length, can be quite large. So we need to make sure that everyone is properly trained to do it. Bloods, well, there are no useful measures of overall macronutrient status. Albumin is not really a reliable indicator of nutritional status in most babies. Glucose, triglyceride levels clearly very important in terms of determining metabolic tolerance. Sodium, inadequate intake, a very common cause of poor growth, often because you haven't fortified the breast milk. Minerals, especially phosphate in the first few days with high amino acid intakes. Hemoglobin and ferritin, very important markers of iron status potentially. We occasionally find the odd baby with low zinc or selenium levels, and occasionally low levels of fat-soluble vitamins. But remember that micronutrient assessment, if babies have acute inflammation, is not reliable. So that's the A and the B. The C, we also need to be thinking about the factors that may affect metabolic tolerance, especially acute inflammation. So during NEC or sepsis, you don't have the same ability to handle nutrients. We need to make sure we avoid overloading the metabolic machinery. Clearly, situations where fluid intake is limited, might need concentrated feeds. Situations where you may have high requirements or increased losses, for example, after NEC. There are very few clinical signs, but you do see babies with poor wound healing, and very rarely you'll see babies with rashes, which may indicate zinc deficiency, but generally these are quite rare. The D bit is that we should all learn how to work out actual macronutrient intakes. You need to do this over the last complete 24-hour period. You multiply your carbohydrate, lipid, and proteins by the Atwater factors, so 4 and 4 and then 9 for lipid. You can quickly work out whether you're approximating macronutrient intakes. More recently, people have started to try and measure levels in breast milk. I think it's quite fiddly. I'm not sure it's really ready for routine practice. But generally, if you're using standardized solutions of PN or milk and you're meeting macronutrient intakes, you're probably going to meet micronutrient intakes okay. And then really emphasizing the importance of the environment and the evaluation, the settings in which we're trying to get these mums to express breast milk or start to breastfeed their babies. The evaluation considering whether the intakes are meeting the needs and whether the babies need more or less, or whether you need specialist device or biochemistry, and then planning with the whole team, and importantly, involving the parents in all that planning. So that's the ABCD schema. I printed it out on a bit of paper here. If anybody wants a copy, you can message me. If you have a dietitian, then maybe you don't have to do this. But I think it's useful for all of us to have a greater awareness of actually how to calculate nutrient intakes. So moving on now, better nutrition undoubtedly is one of the factors that's had a huge impact on survival in preterm babies, and our rates of survival continue to go up. But what this means is that as more babies survive, we're looking after more fragile preterm babies with more complex nutrient requirements and challenges. And when we start to think about why do some of our babies still die, what are the main things that our babies die from? Well, of course, respiratory complications, pretty unusual to die of neurological complications, but sepsis and NEC. And all of these key outcomes now are related to nutrition. So I want just to move on then to, kind of what links nutrition to these outcomes, and how are we starting to think about these things differently. And this is really the bit about gut health and how gut health links nutrition to some of these key neonatal morbidities. Now, NEC is the thing that all of us working on NICUs are absolutely terrified about, and it does tend to dominate a lot of the decision making. And a helpful way of thinking about gut health is perhaps to think of NEC as the tip of the iceberg, with all the other aspects of gut health lurking below the surface. So this is microbiomic, dysbiosis, sepsis, inflammation. So lots of other aspects of gut health that currently seem to be a little bit hidden to us as practitioners, and lots of different ways that we could assess this. So of course, the microbiome, a lot of work on this in the last 10 years, and we're starting to get our head around it, although it's still very complex. But lurking below the surface, what we're trying to do is promote a very rich, stable microbiome. And as the baby becomes more dysbiotic, the risk of NEC increases. But there's also inflammatory process going on. In healthy babies, hopefully very minimal inflammation. But there can be quite a lot of gut inflammation that we're unaware of at the minute because we don't really possess the tools to measure it. You could also think of it in terms of metabolic health or epithelial health. So lots of different ways, but what we need to do is now appreciate that gut health is a spectrum, and it is not a simple dichotomy of NEC or no NEC. Now, when I started doing nutrition, I sort of thought of the gut as this great big long tube that you tip milk into the top end, that you suck out the fluid and the nutrients, and you produce a waste product at the other end. That's kind of what I thought milk was about. I'm embarrassed to say that how naive I was 20 years ago, but I think a lot of other people still think of the gut a bit like this. But what we've come to learn in the last 10, 20 years really is how complex gut physiology is. And we're very interested, and lots of other research groups, very interested in the relationships with nutrients and microbes, immune health, metabolites, and all these kind of things. So as I say, beginning, nutrition is more than simply nutrients, and breast milk is more than food. Breast milk, without doubt, is the most important intervention in preterm infants and intimately related to the acquisition of gut microbes. You're supposed to be born connecting yourself to all the microbes on your mother's perineum and gastrointestinal tract, and then come into contact with the microbes living on her skin or that are promoted by breast milk. And then you're supposed to go home and come into contact with all the different microbes in your environment. And we know now that you have these kind of different waves of microbes. The microbes that come in the first few hours change over the next few days, and that breast milk hopefully promotes this rich, stable microbiota in term babies. And without us seeing it, there's this continual interaction then between microbes and nutrients in the immune system, and the immune system and microbes. That's going on in all of us as we sit here, just as it is in preterm babies. And the relationship and the types of microbes and nutrients changes over the life course. And perhaps an easy way of thinking about NEC is that it's just a breakdown in these three different elements. We know that NEC isn't a single disease. It's the end result of lots of different pathologies. And in some, it may be more due to epithelial factors, and others, it may be more due to inflammatory processes or ischemia. But that, for me, is the easy way of thinking about NEC. And so what we're trying to do is start to unpick, what's actually going on in the intestinal lumen at the top here. What are all these different microbes and metabolites? How does this single layer of enterocytes keep all these harmful pathogens away from the actual tissue? What's lurking in the lamina propria just below the epithelium? We have these dendritic cells that kind of reach out and start to sample some of the antigens in the GI tract, and then feed information to your T cells. And then your T cells interact with your systemic immune system. So we've sketched this out in a recent paper. You could make this type of diagram in lots of different ways and getting increasingly complex. But I think what's really important is to appreciate the huge number of different mechanisms that might be involved and how they're interacting, how what's in the gut, both nutrients and microbes, are interacting with our immune system on a minute-by-minute basis. So here I'm just depicting in a very simplified form this continual interaction between your microbes and your nutrients. And the types of nutrients you put in there lactoferrins, oligosaccharides, whatever, determine the types of microbes that are going to live there. In turn, the types of microbes you have determine your metabolite profile, because these microbes produce short-chain fatty acids, but they also produce amino acids and choline, vitamin K, and all these things, signaling molecules. So there's this continual interaction, two-way interaction. And when it all goes wrong, you end up with NEC. As I say, the causation varies. It's difficult to predict. And this is really where we're struggling in neonatology now, to understand which babies are going to get NEC, how do we predict it, and how do we prevent it. And so although NEC only represents less than 10% of the babies on your unit, we know that it affects our decision-making. Our mechanistic understanding is very limited. Animal models are really not great for this, and there are lots of practical challenges for small babies. But as I say, the way to think of it is that NEC is at one end of a continuum of gut health. And there's no simple or easy measure that we currently have available that allow us to do this. Clinical signs, X-rays are really very crude. And we're all starting to struggle for biomarkers, both in clinical work and research, that may, for example, give us an early warning that gut inflammation is increasing or that the microbial pattern is going wrong, but also biomarkers that help us improve mechanistic understanding. And these could be bacteria or genes or proteins or metabolites, lots of different types of biomarkers that we might have. And we might want to measure them in lots of different fluids. So we might be looking at these in stool, urine, or even blood. So this is a slightly kind of complex diagram. I've almost finished, but I'm just going to kind of go through these different elements. So up here in the top left, I've got things that might be in the stool, top right, things that might be in the urine. This is the epithelial layer here in the middle, and then things that might be in the blood. So different body compartments. And then in green, I'm going to talk about microbial things. In blue, metabolomic things. In red, inflammatory processes. And in orange, things that might be related to epithelial health. And this is based on a great paper from Rusconi in 2018 that I've adapted slightly. So very quickly, just think about inflammatory markers. You can measure these in the stool or the blood. Many of you know about calprotectin. The problem with this is that in the blood, the inflammatory process may not always be specific for the gut. It's quite invasive to take blood from babies. But then if we had some markers in the stool, so for example, RNA or calprotectin, these might be useful measures of gut inflammation. So the first set of biomarkers we want to try and develop would be around inflammation. Next would be microbial aspects. We know that you can get lots of stool from babies, and this might be a great early warning sign, and probably you could develop this into a bedside technology. But there are some disadvantages. The specificity at the minute is not that great. Different NICUs have different patterns, and so what's good for my babies might be different for yours. And we're still not really quite sure which measure of all the different sort of microbial measures we should be looking at. So you can look at markers of epithelial damage, and people have worked on particularly intestinal fatty acid binding protein, which you can measure quite well in the urine, but also other markers of junctional proteins, claudines, and these things. The problem with this is that they're not really an early warning. You only get these markers when the epithelium has been damaged, which is normally a bit late. But urine measures are pretty cheap, and you probably could develop them for use at the bedside. And they might be specific to NEC as opposed to general sepsis. And then really, I think probably the most exciting area coming out of the metabolomic markers. We could measure these in the stool, looking at volatile organic compounds, but also in the urine, and in the blood. And there's been quite a lot of work. I've summarized a few of the studies on the slide there. And if I was going to put my money anywhere and suggest where neonatology might be in 10 years' time, I suspect we will be doing regular metabolomic screening of our babies. I think there is a pattern there that will tell us not only whether the babies are in good nutritional state, but they're going to tell us something about gut health and about inflammatory processes and things like that. We're not there by any means yet, but I think that that's potentially where we might be going. So here really I've put some of these biomarkers together and just shown you that whilst lots of these biomarkers are abnormal once you've got NEC, what we're really searching for are biomarkers in the very early stages and the babies are still healthy, trying to work out whether the baby is at higher risk of developing gut complications. So I think we're moving into this area where we're trying to integrate nutritional status and gut health into clinical practice. Lots of complex nutrient metabolite interactions. Very important that we understand how to promote healthy epithelium, how we feed the brain. And I guess we're getting to the stage now where we're asking the question, are we ready for prime time? Have we got to the stage where we understand enough to actually put this into practice? So as well as the ABCD, where is it that we might be going? And I think probably in the immediate next few years, we need better measures of linear growth and body composition at the bedside. We can start working on some of these biochemical measures to refine them. We certainly need to be much better at integrating electronic data and integrating the stuff that's already collected at the bedside there. And as I say, thinking about those blue elements of nutrition, working better with parents to promote breastfeeding and support them. And this is what things might start to be looking at. So there are now techniques to measure the microbiome at the bedside. You can measure the 16S profile and stick it in a USB into your computer. Here in the middle, I've put this Metabolomic and eNose thing. So all of us go through airports, we did before COVID stopped us flying. And every now and again you get stopped and they take a swab from you and they put it into one of these machines. And this is just basically pattern recognition. That machine doesn't tell you what it is you're carrying on your hand. It just tells you whether the pattern looks abnormal, whether that pattern looks like the pattern you might see associated with explosive residues. And I think that there is hope that you might start recognizing these patterns either in the baby's stool or in the urine. Maybe we're going to start analyzing breast milk a bit more. This is data that we haven't yet published, so I can't tell you what it's in relation to, but these are HMO profiles in maternal breast milk. You see these ones here on the left with blue are from secretor mums, and these are from non-secretor mums, but we're starting to understand that the pattern of HMOs might predict your risk of disease. Here are some really cool diapers where you can actually just implant some chips into them and they'll turn a different color. I think this one was really just to tell you whether you needed to change the nappy or not. But just imagine if you can have one of these and it started going red if the baby looked like he might be developing NEC. And then we need to really work on the microbiome and understand the different transitions that happen between the communities. What we know is that the pattern of NEC is not with a single microbiome pattern, but the rapid shifts and instability are probably the thing that increase the risk. So almost finished. We are getting better. This is survival from our unit over the last 15 years. And you can see that at every gestation, survival is improving. But these babies only get given one brain. An outcome is much more than simply survival. We need to better understand how to protect and nourish that brain. And better understanding nutrition is clearly key. So this is where we've got to. We've done the ABCDE. Perhaps we're now starting to move into the F, fecal and urinary analysis, and get a better understanding of gut health and point of care omics. And I'm going to finish there. Just highlight again that if you want to copy these slides, you can get them from Astati or you can go to our website. And I think I've finished with 10 minutes to spare and happy to take any questions. So thank you very much.
All right. Great. Thank you so much. I'm going to make you big on the screen. And, we've got some questions. Okay. Do we need to be concerned about rapid catch-up growth in preterm infants?
Not in the very small babies. I think there's no doubt that rapid growth in the first few weeks and months after term in normal term babies may well be harmful for metabolic health. It may be worse in term babies who are growth restricted. But in preterm babies, the overriding focus of neonatal nutritional care has to be to promote good brain growth. So if the babies are less than 32 weeks, please focus on feeding that baby's brain. You can worry about their obesity or their risk of diabetes. My 24-weekers would be delighted to be alive when they were 50 and be on antihypertensive medicines. So you need to feed them so that they can actually survive to get their diabetes. So honestly, I don't think we need to be worried about rapid growth in these very small babies. Whether you need to be worried about it in the 34, 35 weeks, I don't know. There isn't very good evidence that nutrition at that stage has a big impact.
Great, thank you. The next one, how do you distinguish NEC from spontaneous intestinal perforation?
You can only do that at surgery. It's very difficult on a abdo film to know for certain whether the baby's got NEC or perforation, but if the baby's got a perforation there, the baby needs an operation regardless. Of course, there are differences in the presentation. Spontaneous intestinal perforations in the terminal ileum tend to happen in the first few days. NEC tends to be a bit later. We are starting to see some slightly different types of perforation. We're starting to see some mid-ileal perforations, which are a bit bigger than the classic ischemic ones. So there's some different pathologies going on there. But the really take-home important message is speak to your pathologist, make sure that every baby who goes for an operation gets good histology, and review that histology and make sure you code those babies correctly in your database. Because if you're coding them all as perforations and not NEC, then you're never going to work out whether probiotics are useful or not.
Great, thank you. The next one, what do you think about SMOF? You said something about it won't be the best option for premature babies. We have been using it with great answers from babies in terms of preventing cholestasis.
Yeah, so SMOF, which is a lipid, I don't know how widely available it is in the US, but it's widely available in Europe. It's made from fish oil, olive oil, and fungal sources. It clearly has a very different blend to what you would have in the old soybean lipids. There is absolutely no evidence that using SMOF or these third generation lipids routinely in preterm babies improves outcome. No evidence of that at present. They just need the lipids for the essential fatty acids and the calories. Whether SMOF is better for babies who get stuck on PN long term hasn't been fully determined. We definitely think that we see less cholestasis with SMOF compared to intralipids. So we don't see cholestasis in the short gut babies now compared to what we saw maybe a few years ago. But don't start assuming that SMOF is the answer to everything. It's not a lipid that was designed for preterm babies. The blend just isn't right. You have much higher levels of icosapentaenoic acid, and there may be all kinds of other things that we're doing harm to these babies. And there are even concerns that some of the inflammatory metabolites produced from SMOF might be harmful to the lungs. So we do use it. I think it's probably better than a soybean oil preparation, but it's by far from a perfect lipid for babies.
Okay, great, thank you. Next one, feeding during transfusion and impact in nutrition if withholding feedings.
Yeah, hot potato that's been doing the rounds for many years now. The only way to resolve this is to do a really big trial. There is a big trial planned in the UK, but you're going to need numbers in the order of sort of 2,000-3,000 babies to do it. I don't have a strong feeling. I'm personally not sure that there is an association based on the overwhelming epidemiological data. To me, it doesn't tell me that it's transfusion that's doing the harm. Possibly the causative or the etiological factor is more associated with the anemia rather than the transfusions. So whilst we don't want to give babies lots and lots of transfusions, we do need to stop them getting anemic, because then they won't perfuse their gut, and that might be a risk factor. But in answer, I don't stop feeds currently, but I would put my babies into a trial because I have equipoise. I honestly don't know the answer to that at the minute.
Okay, great, thank you. Next one, what are the common nutrients lacking in standard preterm diets?
Well, there's a little trick question here, which is what are the important nutrients for babies' brains? And the answer is every single nutrient is important for a baby's brain. If you gave every nutrient perfectly, but you missed out on just one, it would be totally wasted. So human metabolism requires all your vitamins and cofactors, micronutrients and macronutrients. You need all of them. Otherwise, they wouldn't be there. So I think in general terms, we need to sort of, just understand that everything's important. What are the common things that go wrong? The common things that go wrong are people don't give enough macronutrients early on because they can't add up the numbers, or they don't know what to give, or the babies won't tolerate it, or things like that. So macronutrients is the really big one. Sodium can be an issue sometimes in babies. It drops low and people forget to keep a check on it. Phosphate, very important in the first few days, particularly with higher intakes of amino acids. So keep a check on that. And then later on, remember iron. But you won't become anemic due to iron deficiency. If you're anemic due to iron deficiency, you've missed the boat. You've waited too long. So don't let them get iron deficient, because you will be harming the brain.
Great, thank you. The next one, does slow growth really matter if the baby is well?
Yep, complex question. If the baby genuinely is really well, we need to appreciate that there's a variation in normal, and some babies will grow at a slightly slower rate to others. So we shouldn't expect all babies to grow at 20 grams a kilo a day at 25 weeks. Some just won't do that. What you need to do if you see slow growth is make sure that the slow growth isn't due to inadequate nutrition. If you're growing slowly because you're not giving the baby enough macronutrients, then yes, slow growth is a concern. If you're absolutely sure that you're meeting all the nutrient requirements as best you can, and particularly you're optimizing the amount of mum's milk you can get into the baby, and as I said earlier on, the one thing that we're all agreed on is that the amount of mum's milk you get into the baby is the one thing that really makes a big difference short and long term. So if you've maximized, optimized absolutely everything you can, and the baby tends to be growing slower than normal, that's probably just what the baby wants to do. So we shouldn't ignore it. But as I said before, we're not trying to achieve growth. We're trying to achieve good nutritional status, and we're trying to grow the baby's brain.
Great. Can you comment on use in babies under 32 weeks versus other gestational ages?
Probiotics. Yes. That's a tricky one. That probably would take another whole hour to talk about, or maybe even a whole day. My own feeling is that the overwhelming evidence supports the routine use of probiotics if you can get hold of a preparation that you're happy is of high quality. However, I don't know which of the preparations you should be using. I'm not sure we know exactly which strains and which combinations you should be using. As part of ESPGHAN, Chris Van Acker did do a network meta-analysis, so we have an idea, but we only have an idea of what's available and what seems to be better than some of the other ones. We really don't know yet the perfect combination, and we need to get a better understanding of how probiotics interact in the gut. What is it that they're doing to all those different elements of gut health? Are they suppressing inflammation? Are they promoting a more healthy microbiomic mix? I don't think we know yet.
Okay, great. Is there any proof that human milk fortifiers are better or safer than bovine-derived fortifiers?
Yeah, that's a very kind of tricky topic. I've declared a conflict, which is that I have done a study with Prolacta, but we haven't analyzed all the data yet. My take is that the data look very interesting. There are a number of small trials and some observational evidence that suggests there may be a benefit in avoiding bovine products. But we know that to really nail the question, if what you're interested in is NEC or sepsis, we know that to nail that question, you need a big trial. You're probably talking 1,000 babies, and that trial currently doesn't exist. So I think it's up to individual units to look at the data and decide whether they feel the data is strong enough. The way that I view it currently is opportunity cost. I'm not denying that if everything were equal, people might want to opt for a human milk product. But these products are not cheap, and therefore the money that we have to spend can only be spent once. So if you're going to spend it on human milk products, then that you can't spend it on something else. I think you need to decide whether that's really where you want to put your money. I couldn't make a call on that at present.
Okay. Comment on nutrients that you would add when baby's receiving donor breast milk that's different from baby receiving mother's own milk.
Yeah, I personally don't really draw a distinction because although whether you're on mum's milk or donor milk, if you're less than 32 weeks, you're going to need a fortifier to meet your nutrient requirements pretty much. And we know that with donor milk, you're going to probably need higher levels of macronutrients. I think a lot of donor milk banks now do provide you with an analysis that you perhaps could titrate your fortifiers, but the fortifiers aren't really designed for you to fiddle around yet. So our supplementation at the minute is pretty crude. I mean, I agree with the reasoning behind the question, which is to say, is donor milk, are donor milk nutrients the same as mum's milk nutrients? And they're not. They tend to be very different in quality, and they tend to be lower in density. So yeah, we probably could do with a more refined approach to fortification than we have at present.
Okay, great. And I'll ask one more question, and then we're at the end of our time here. How much will a better understanding of gut microbiome improve care?
Well, I think we're all starting to realize, not just in preterm babies, but in general, how important the gut microbiome is to health at all different stages. I mean, when you start looking at the literature, it's absolutely overwhelming how the microbiome relates to all these diseases that you wouldn't have imagined to have anything to do with it. Data to show that your microbiome differs if you have various psychiatric conditions, different patterns in the microbiome of people 10 years before they develop clinical signs of Parkinson's. I mean, it is really quite amazing how important the microbiome, the gut microbiome, tends to be for all these different diseases. Where we're stuck at the minute is that all these huge and interesting studies are all largely observational. And are we really looking at causation, or are we looking at association? Now, that may not matter for a biomarker. I mean, if it's abnormal, it's abnormal and it might predict disease. But when it comes to mechanisms, we currently don't really know whether the abnormal microbiome is driving the pathology of gut inflammation, or whether some other process that's driving inflammation is also driving the microbiome. So is the microbiome a marker and an innocent bystander, or is it really on the causal pathway? And yeah, we're not there yet. Thankfully, time for a bit more research.
Okay, great. Well, I want to thank you so much for your time today and for all this great information, and look forward to seeing everybody at our next webinar on September 17th with Dr. Catherine Gregory. So thank you so much, Dr. Embleton.
Bye, everybody.
Later. Bye.
Bye.